Hello everyone. Today we are going to read about the uh, Kerbs cycle or citric acid cycle. This is also called Tca cycle. So the Tca cycle commences after the formation of the acetyl enzyme by the action of the enzyme pyruvate dehydrogenase on the substrate pyruvate. So the major production of the ATP, like 65 to 70% of the ATP is produced from the citric acid cycle, and this citric acid cycle specifically occurs in the mitochondria of the cells. So those cells who are lacking the mitochondria, like RBC, lens, cornea, there is no any occurrence of the citric acid cycle. Those cells who only had the mitochondria, um, they in those cells only the citric acid cycles occur. So now let us discuss the citric acid cycle and the ATP production in the citric acid cycle, and the regulation of the citric acid cycle. So now let's start. So from the glycolysis, the pyruvate is formed. So from glycolysis, from the glucose molecule, the pyruvate is formed. How it is formed? I want you to read the detail about the glycolysis cycle. If you don't know that, I request you to read that how glucose molecule is converted to pyruvate by the process called glycolysis, and the pyruvate is converted to acetyl coenzyme by the pyruvate dehydrogenase enzyme complex. So this the pyruvate dehydrogenase enzyme complex. I have also already discussed about this pyruvate dehydrogenase in detail in my previous video. So if you haven't learned that, if you don't know that, I want you to read detail about pyruvate dehydrogenase before reading this Kerbs cycle or citric acid cycle. So we have the molecule, the acetyl coenzyme. We have the molecule acetyl coenzyme. Upon the action of the pyruvate dehydrogenase enzyme on the pyruvate, the acetyl coenzyme A is obtained. So the oxaloacetate, which is 4-carbon compound, oxaloacetate along with the acetyl coenzyme A, with the help of the citrate synthase, the citrate is formed. The citrate is 6-carbon compound, and from the citrate the cis aconitate is formed by the action of the enzyme aconitase with the production of the water. From the cis aconitate the isocitrate is formed. Here also cis aconitase acts upon the cis aconitates. Aconitase acts and isocitrate is formed along with the production of the water. The isocitrate is also the 6-carbon compound. So on the isocitrate dehydrogenase acts, forming the oxalosuccinate, and again the isocitrate dehydrogenase acts on the oxalosuccinate to form the alpha ketoglutarate. The alpha ketoglutarate is the 5-carbon compound. So you have to know this. So from the isocitrate, the 6-carbon compound, the isocitrate dehydrogenase acts to form the alpha ketoglutarate. So on the alpha ketoglutarate, the alpha ketoglutarate dehydrogenase enzymes act along with the help of NAD plus, forming the NADH plus H hydrogen ion, and also forming a compound called succinyl coenzyme A, which is the 4-carbon compound. On the succinyl coenzyme A, succinate thiokinase enzymes act, forming the succinate. So in this, the GDP plus phosphate ion also act, forming the GTP and COASS coenzyme A. So I want you to know that here in this reaction, upon the conversion of the succinyl coenzyme A to succinate by the action of succinate thiokinase, GTP is produced. So this reaction is the substrate level phosphorylation. I have already told you in many videos previously that when a compound form ATP without the involvement of the electron transport chain, then it is the substrate level phosphorylation. There is automatic generation of the ATP from the substrate, then it is substrate level phosphorylation. So this GTP acts with the ADP, forming the ATP and GDP. So from here, from this GTP, the ATP produced like this. So we have the succinate compound, which is 4-carbon compound, and from the succinate dehydrogenase enzyme acts upon the succinate along with the production of the FADH2 with the help of FAD. Fumarate is produced. The fumarate is 4-carbon compound. On the fumarase acts upon the production of the water. The malate is produced, which is the 4-carbon compound, and from the malate oxaloacetate is produced, and in this reaction NADH is also produced by the help of NAD+. So from the malate oxaloacetate is produced by the action of the malate dehydrogenase, and there is production of the NADH. So this cycle goes on and on and on. So now let us discuss about the ATP production in the citric acid cycle, this Kerbs cycle. How much ATP is produced? So in the citric acid cycle, 3 NADH is produced. So I want you to see 1 NADH is here. This is the 1 NADH. Another 1 NADH is here on the enzyme alpha ketoglutarate. Another NADH is here on the enzyme malate dehydrogenase. So 3 NADH is produced and 1 FADH2 is produced. So here you can see 1 FADH is present in the succinate dehydrogenase enzyme on the reaction of the succinate dehydrogenase enzyme. So 3 NADH, 1 NADH is on the, when there is conversion of the isocitrate to alpha ketoglutarate, 1 NADH is produced. Another is when there is action of the alpha ketoglutarate on the substrate, on the molecule alpha ketoglutarate, when it is changed to succinyl coenzyme, another NADH is produced. And last, when malate is converted to oxaloacetate, then another NADH is produced. So 3 NADH are produced and 1 FADH2. While there is reaction of conversion of the succinate to fumarate by the action of succinate dehydrogenase, 1 FADH2 is produced. And 1 GTP is produced. 1 GTP is produced when succinyl coenzyme A is converted to succinate. 1 GTP is produced upon the action of succinate thiokinase. So, according to new study, 1 NADH produces 2.5 ATP, whereas 1 FADH2 produces 1.5 ATP. So this 3 NADH goes into electron transport chain. 1 FADH2 also goes in electron transport chain. That means 3 into 2.5, 7.5 ATP. 1 FADH2 plus 1.5 ATP plus this 1 GTP is converted into 1 ATP plus 1 ATP. That means 7.5 plus 1.5 plus 1 is equal to total 10 ATP is produced per acetyl coenzyme A molecule. So total 10 ATP is produced per acetyl coenzyme A molecule according to new study. Now let us discuss about the regulation of the citric acid cycle. So mainly the regulation of the any reactions occurs only in the irreversible reaction. In the reversible reaction, mainly there is no any regulation. So first regulation we see while there is formation of the citrate by the action of the oxaloacetate, acetyl coenzyme A and citrate synthase. The ATP inhibit the action of the citrate synthase, and if there is more production of the ATP, that means there is more availability of ATP, it inhibits the enzyme citrate synthase, so that citrate wouldn't be produced and Kerbs cycle wouldn't be commenced. So mainly if there is more availability of ATP, the Kerbs cycle would be less. Another is The action on the isocitrate dehydrogenase, the ATP and NADH inhibits the enzyme isocitrate dehydrogenase, and ADP uh, activates the enzyme isocitrate dehydrogenase. Also, the alpha ketoglutarate dehydrogenase is inhibited by succinyl coenzyme A, NADH, and ATP. You have to know this clearly. So you have to think like that. Mainly the Krebs cycle or citric acid cycle is required for the production of the energy. The energy currency in our body are ATP, NADH, FADH2. So if there is more availability of the NADH, FADH2 or ATP, automatically the Krebs cycle would be less. You have to think like that. You have to think logically. So in the MCQ, you just need to know that ATP inhibits the citrate synthase, ATP NADH inhibits the isocitrate dehydrogenase, whereas ADP increases the action of the isocitrate dehydrogenase. Succinyl coenzyme A, NADH, and ATP inhibits the enzyme alpha ketoglutarate dehydrogenase. And the substrate level phosphorylation also occur in the Krebs cycle, while there is conversion of the succinyl coenzyme A to succinate by the action of the enzyme succinate thiokinase. This is the main thing you have to know in the citric acid cycle or Krebs cycle. So we have learned about the detailed cycle, how these cycles occur. We also learned about the ATP production in the citric acid cycle. We also learned about the regulation of the citric acid cycle, how the citric acid cycle is regulated. I hope you understood this lecture, and I want you to make the very good notes of the various cycles of the biochemistry that you should know, like glycolysis, Krebs cycle, and other various cycles I will discuss in detail, so that you will not forget, so that you will revise it regularly, so that you can revise regularly after making your notes. So these cycles are very volatile in the memory. You have to revise again and again to memorize all the stuffs like this biochemistry cycle. So I hope you will do better, and I hope you all the best for your MD examination or various undergraduate examination. Thank you.